New trends in plant breeding. Marker-assisted selection. MAS is a technique that uses molecular markers linked to desirable traits to assist in the selection of plants with superior characteristics. Traditional breeding involves phenotype-based selection, which is time-consuming and less precise. MAS offers a more precise and efficient way to select specific traits. MAS uses molecular markers, specific DNA sequences like simple sequence repeats, SSRs, and single nucleotide polymorphisms, SNPs, that are associated with desirable traits, allowing breeders to select plants with favorable genetics more efficiently. These markers enable precise identification and selection for key target traits, including disease resistance, yield enhancement, quality improvement, and stress tolerance, which are vital for crop resilience and productivity. Genetic linkage plays a crucial role, as markers closely linked to genes controlling these traits are identified and used to track and select plants with the desired characteristics during breeding cycles, thereby accelerating genetic improvement and minimizing reliance on traditional phenotypic assessments. Simple sequence repeats, SSRs, also known as microsatellites, are short, repetitive DNA sequences, typically 1 to 6 base pairs in length, that are scattered throughout the genome. These repeats are highly polymorphic, meaning they vary significantly in the number of repetitions among individuals within a species, making them valuable markers in genetic mapping and breeding. SSRs are particularly advantageous in gene mapping because they are codominant markers, which allows for the differentiation between homozygous and heterozygous genotypes. This is critical in breeding, as it helps breeders identify plants carrying beneficial alleles in a single generation. SSRs are also highly reproducible, easy to amplify via PCR, and stable across generations, allowing consistent tracking of traits over multiple breeding cycles. Their high variability and widespread occurrence across the genome make SSRs ideal for evaluating genetic diversity, identifying QTLs, quantitative trait loci, linked to key traits, and implementing them in marker-assisted selection, MAS, for crop improvement. Single nucleotide polymorphisms, SNPs, represent the most common type of genetic variation, occurring as single base pair changes at specific positions in the DNA sequence. They are valuable as molecular markers because they are highly abundant, evenly distributed throughout the genome, and often associated with genes influencing phenotypic traits. SNPs provide high resolution for mapping genes and QTLs, as even single base variations can be traced in linkage disequilibrium with traits of interest, offering breeders precision in tracking inherited traits. SNP are amenable to high-throughput genotyping, enabling the screening of thousands of SNPs in large populations, which enhances the efficiency and accuracy of MAS. SNPs have become integral to MAS, especially for complex traits like yield, disease resistance, and stress tolerance, where fine mapping of genes is necessary to develop resilient and productive crop varieties. SNP markers, due to their abundant presence and high specificity, are now widely applied in modern crop improvement programs to streamline the identification and selection of desirable alleles in breeding populations. Marker-assisted selection, MAS, involves a systematic approach to plant breeding that enhances the efficiency and accuracy of selecting desirable traits. The process begins with marker identification, where researchers locate molecular markers that are closely linked to traits of interest, such as disease resistance or high yield. Following this, genotyping is conducted, which involves analyzing the DNA of various plants to detect the presence or absence of these specific markers. Once the markers are identified, the selection phase involves choosing plants that exhibit favorable marker profiles, ensuring that the desired traits are more likely to be passed on to the next generation. Finally, validation is essential to confirm that the identified markers are reliably associated with the target traits, ensuring the effectiveness of the selection process. This validation step often involves additional experiments and trials to verify the accuracy and stability of the marker-trait relationships. Together, these steps enable breeders to make informed decisions, accelerating the development of improved crop varieties with enhanced performance and resilience. Advantages of MAS. Increased precision selection is based on genetic markers, not just physical traits. Speed, faster than traditional phenotypic selection methods. Non-environmental influence, traits linked to molecular markers can be selected even in non-ideal environments. Complex traits, facilitates the selection of traits controlled by multiple genes, e.g., disease resistance. MAS in disease resistance breeding enables the early identification of plants with specific resistance genes, such as the XA21 gene in rice for bacterial blight resistance. This approach accelerates the breeding of resilient varieties, reducing disease impacts on crop yield and ensuring more stable production. MAS in abiotic stress tolerance enables breeders to identify and select plants with traits for resilience against drought, salinity, and temperature extremes, such as salt tolerance in barley or drought tolerance in wheat. This approach supports stable crop productivity in challenging environmental conditions, helping to secure food supplies amid climate stress. Challenges in MAS include limited marker availability for some traits, high costs associated with genotyping, and the complexity of selecting multi-gene traits, which may need advanced marker systems. Future advancements in marker technology and cost reduction could expand MAS applications in plant breeding. Thank you for your time.